Good afternoon, everyone. My name is Justin DiAngelo, and I'm going to start us off today. So as Rachel said, there's four of us. We are all authors of this paper, and our names are listed here on this first slide. So we're going to go through each of us one at a time to introduce ourselves and talk a little bit about our background and how we got into biology education research. And then we will go through our presentation. Feel free to ask questions throughout, and we will stop as we're going through if we feel we can answer your questions at that point. And if not, we'll get the rest of the questions at the end of the session. So as I said, my name is Justin DiAngelo. I'm a faculty member at Penn State Berks, which is one of the undergraduate campuses of the Penn State system. And I've been a faculty member there for about 5 years, and I teach biochemistry and molecular biology. And I got into biology education research through the Genomics Education Partnership, which is what we'll talk about here in the context of this paper. And so while I have designed cures and other sort of active learning exercises for my courses, my main assessment of education research has been through the GEP.
Hi, everyone. I'm Amy Hark. I'm a professor of biology at Muhlenberg College, which is a liberal arts institution with about 2,200 undergraduate students. I started paying attention to science education, research, and literature as sort of professional development when I was a grad student. And I've engaged in different kinds of scholarship of teaching and learning throughout most of the time I've been at Muhlenberg, so for about the last 15 years. I've published reports on service learning, on course modules, I've developed case studies, but participating in the GEP has really allowed me to be part of a larger science education effort.
Hi, everyone. I'm Ann Rosenwald. I'm at Georgetown University. We're an R1 university, and we have about 5,000 undergraduates. I'm in the biology department, and I got involved in science education back in about 2004 with Malcolm Campbell's GCAT program. And I'm currently a member not only of GEP, but also another related group called Genomics Education Alliance, and another group called NIBBLS, which stands for the Network for Integrating Bioinformatics into Life Sciences Education. And I also have been the PI of a smaller project that I work on with two of my former postdocs now called Genome Solver. And I'll put in a plug. We're doing a Genome Solver workshop in December. If anybody wants some basic bioinformatics tools, you can let me know.
Matt Wawersik at William & Mary, a small public research university in colonial Williamsburg, about 5,000 undergraduates. My research focuses on developmental genetics using Drosophila, and I've been involved in this GEP group doing science education work since about 2008. And similar to Justin, it was really that's my primary place where I've been doing science assessment. But it's really been allowed me to develop some cool lab experiences. A lab that I teach is a genomics and functional genetics lab that I use for the assessment and is incorporates GEP materials. So turn it back to Justin.
Okay, so I'm going to give you all a little bit of background about the GEP, or the Genomics Education Partnership, and then frame the scientific question we were posing in the paper that was published in GIMBY. So the GEP was started back in the mid-2000s by Sally Elgin, who was a faculty member at Washington University in St. Louis. And she received an HHMI professor grant to form the GEP with the goal of incorporating genomics and genomics research experiences into the undergraduate curriculum. So Sally essentially ran the show up until 2017. She recruited members, helped organize training, came up with the scientific projects, and organized all the assessment efforts. So the scientific projects broadly all revolve around annotation of different of interesting genes or regions of genomes. And so it started focusing mostly on Drosophila because Sally is a Drosophila geneticist, and she worked on the F element and epigenetic questions. So we did mostly annotated Drosophila F element regions. But more recently, we've expanded the scope to include additional Drosophila projects with various signaling pathways in Drosophila, as well as wasp venom genes, parrots, right? We're expanding lots of different genomics projects based on the interests of members or scientific partners. So speaking of members, we have close to 200 members at a wide range of institutions. So the map on the right-hand side of the slide here shows all of our members. All of our members are indicated by a dot. And so you can see that we have a wide geographic range, but we also have a wide range of types of institutions, all the way from teaching and focused institutions to R1s. So over the past few years, once Sally announced to us that she was going to retire, right, we have transitioned the GEP to a more distributed leadership model. So our current leader, scientific lead and program director is Laura Reed at the University of Alabama, but she can't do it all. So we have designed a number of committees that are shown on the right-hand side of this slide to essentially distribute the load of getting all the work done. So, we have folks who are focused on the science that we do, some folks that are focused on the training aspects, developing curriculum, and then of course the assessment piece, which is really important for all of our biology education studies that we do and what we're going to talk about today. And the advantage of having such a large group of faculty involved is that we can really ask some interesting and unique education questions using this large sample size. Okay, so for the paper that we're going to discuss today, our overall hypothesis was that like most research endeavors, right, the students that are involved in our research experience, they undergo some frustration, like most of us do. But that frustration aids in student learning. And we specifically were focusing on a CURE setting, right, a classroom undergraduate research experience. And so, we know by doing annotation that annotation can be hard. Okay, so if you're not familiar with annotation, right? What annotation is taking sequence of DNA and trying to determine the overall structure of any genes that may be found in that DNA, right? Where are the introns, the exons, the splice junctions, et cetera. So we use a lot of evidence in order to try to determine this. And so on the right-hand side of this slide, I have a screenshot of the UCSC genome browser, which is sort of the workhorse piece of software that we use through the annotation process. And each row here is a different piece of evidence. So the students have to go through all of this evidence, some of which is consistent and some of which is not consistent with each other, right? And they have to use what they know about genes and genomes in order to determine what the best gene model is. And this can be a challenge, okay, for them and for us. And so this idea of frustration aiding in student learning started with anecdotal evidence. So we have a yearly workshop or get together where members of the GEP come back to St. Louis and we, share our experiences and do work that's important for the GEP. And so we would have these conversations where we heard a lot of people say that there was a lot of frustration, but students worked through it and it was really good for them. So we looked into the literature, and lo and behold, there were a number of examples of this, not only in the biology literature, but literature from a number of science and social sciences and different contexts. But never were they looking at a sort of cure or classroom undergraduate research setting. So we set out to address this hypothesis, and we wanted to look at a number of different data sets and try to triangulate the data, which triangulate's one of our favorite words, right? Because we wanted to look at different sets of data, like faculty and student surveys, as well as some some student focus groups, to help to see if we could capture any of this frustration leading to student learning in a very objective way. All right, so now I'm going to pass this on to Amy, I believe.
Thanks, Justin. Thanks, Anne, for moving the slide forward. Yes, I'm going to talk about one of the approaches that we employed in this study. And this did stem out of conversations at that alumni workshop that Justin was talking about. A smaller group of us was in a breakout session, and we were talking about the fact that we really felt that when students struggled initially in these GEP, gene annotation projects, it was actually beneficial for their overall learning. And so one thing we wanted to figure out in exploring this further was whether GEP faculty across the board, from different institution types with different student populations, whether they shared that same observation that this frustration was helpful for learning. So we developed a survey and circulated that for editing. And then we shared that out and asked all GEP faculty to complete that survey. The questions on the survey were open-ended and the comments were anonymous. So we didn't track who said what. And then we analyzed this using a quantitative analysis software called NVivo. And our goal was to capture themes and sort of arrange the comments into categories. And Anne, if you can flip to the next slide, then we can talk a little bit about the results from the faculty survey. So to summarize these results, when faculty were asked to report an obstacle that they encountered sort of in executing this cure, this classroom undergraduate research experience, one theme was challenges that had to do with course structure, limits to time, challenges of technology. And another theme was around sort of student issues. And the common challenges that faculty reported that they found students having had to do with situations where students were confronted with a situation where the gene annotation, the model they were building, wasn't following the rules they expected for molecular biology that you would learn in a textbook. So a non-canonical splice site, a non-canonical start codon, things like that. And another student challenge that faculty reported was students trying to integrate all these multiple sources of data that Justin was showing in the genome browser, right? How to put those together to build a model. So faculty reported those obstacles. And then in terms of reporting how students overcame those challenges, the most frequent thing faculty said was that students changed how they were working on these problems. And notably, the primary thing that faculty reported was that students were increasing their collaboration, either with other students in their course or working with the TA in situations where a TA was available or with the instructor. The third point here in this results slide is that when faculty were asked about how this impacted students, students' initial responses to these struggles, to these challenges, the obstacles, could be seen as negative. But faculty reported they felt those initial, maybe negative reactions were offset by increased engagement and increased learning, both in terms of the subject matter, genomics, but also in terms of the research process. And if you can just flip to the next slide, we can maybe just look at a few examples of data from the paper. So figure 2, right, reports how faculty felt about how course setbacks on the left impacted student learning and how student challenges impacted student learning. And particularly notable for the student challenges, faculty actually overwhelmingly, the majority of faculty felt that those challenges were actually beneficial or positive for student learning. Positive comments are the blue bars in this graph, negative are orange, and no effect is gray. So really seeing a lot of positive outcomes reported by faculty in terms of what happened with course setbacks and student challenges. And then the quote on the right of this slide is just one representative quote that's in table 2 in the paper. I'll just read it quickly. This is a faculty member talking because this is the faculty survey data. Both the students and I felt that they really had achieved something when they were eventually able to get the gene feature correctly annotated. They expressed a strong sense of accomplishment when it finally, quote, worked, and clearly stated in the end it was, quote, worth it. So this is just some sample data from the paper on the faculty survey. And now I'm going to turn it over to my colleagues to talk about the student survey and other student comments.
So our question here was whether or not the student comments reflected these faculty observations. So to use Justin's word, triangulate, could we see evidence of what Amy was talking about in what the students said? So our methods here for the student survey comments, the students are asked when they're involved in a GEP experience to participate in a post-course survey. And so we asked for comments on the post-course survey, and these comments were solicited with a really broad prompt. We didn't ask them specifically, were you frustrated? But we looked for evidence of that word in there, in this prompt. Please comment on the effort made to integrate research and teaching. What were the strengths? What were the weaknesses? What was of special value to you? So over the 2 years of data collection, we got 647 comments in response to the prompt. I mean, one of the other aspects of working in a big group like this is not only do we have 200 faculty members, but each one of those faculty members has many students. So we have a big data set. We also analyzed this work with in vivo, looking both for emergent themes, and then we also fed it keywords and stems that were associated with the idea of frustration. And so we had the computer do it first, and then we had human people look at these keywords to understand the context, because we were looking for things that would say frustration and success, but clearly something like not successful would not be something that measured success, right? So we were looking for kind of stricter criteria there. So the emergent themes that came out of the student survey comments, we had a number of positive themes. They liked the classroom atmosphere. They were motivated. They liked the learning. They understood science better. They had some better ideas about career clarification. There were negative themes, time issues. The task is hard and it's uncertain. But what struck us as we initially looked at these was a lot of them had both aspects in. And so that's kind of where we came up with this frustration through to success idea. So the key words, we used words like this: frustration, overwhelm, struggle, burden, knowledge, learning gains, understand, confidence, success, appreciation of the scientific process, scientific method, hypothesis, evidence, things like that. So when we put all that together, about 20% of all the 647 comments said something about frustration. And here's an example. It was extremely frustrating and confusing to understand. And again, remember, we didn't ask them specifically, were they frustrated? But of those comments that mentioned frustration, more than half also mentioned success. Huge learning curve. I almost gave up, and one day it clicked and I was able to finish the project. Overall, this class was challenging, but very rewarding once you had those moments. So that's kind of what we're looking for, right? From our students. We want them to persevere through challenges and come out the other side. So that was kind of interesting to see that we could see that in these comments from the students. So now I'll turn it over to Matt. He's going to talk about the student focus groups.
And I have unmuted, so we're good there. So yeah, in our efforts to triangulate with the data suggesting that frustration is leading to success, we not only wanted to look at the student surveys, but also student focus groups. And so what we did was to assemble 7 groups of about 6 to 8 students from a diverse range of schools. There was one R01 institution, 2 state universities, 3 four-year colleges, and 2 of these colleges were actually minority serving institutions. And so for these groups, there was then a independent facilitator that asked a series of scripted questions to the students. And these questions had sort of umbrella topics under the context of understanding science, career trajectory, or personal gains, not necessarily looking for frustration or anything, as Anne was talking about in the student surveys, but then the answers, and there were 669 of comments from this conversation with a facilitator, were then plugged into this algorithm keyword analysis software called NVivo. And Anne talked about some of the keywords that were looked for. But then from the keyword analysis, there was a secondary analysis done, both looking for overlap between this concept of struggle that might have come out, as well as success and various gains. And this secondary analysis was done both by the keyword algorithm as well as by humans manually. And so next slide. And so the primary analysis, not looking for any overlap, showed a high number of references to learning gains, enhanced understanding of the scientific process, as well as obstacles and frustrations. So similar key things came out from the NVivo analysis in the student surveys. And then digging in deeper to look for overlap, which is the key thing, we looked for, again, overlap between situations where an obstacle was linked to learning gain or enhanced understanding of the scientific process. And the manual analysis actually showed that 72% of the comments referencing struggle also referenced learning gains or better understanding of the scientific process. And the secondary NVivo analysis showed similar results. And so just some representative quotes from this showing in particular struggles leading to benefits in understanding of the scientific process. The first is we had troubles and we always approached it from different angles because you have to. And it just made you want to explore it more, right? So there's this struggle leading to success. And another quote, I think this was the best way to learn because you are not passively observing. You're figuring out reasons why it has to be the way it is through trial and error. And this combination of trial and error and different angles really are, I think, important points with the iterative process and looking at things from different perspectives in the scientific process. And so together, if you look at all this evidence, whether it's the faculty or student comments, all the data triangulates on this idea of allowing students to struggle enables success. And those successes aren't necessarily just enhanced topical knowledge, but less tangible things like better understanding the research process and questioning assumptions, which is huge for budding scientists. And so this is, the data shows real value just beyond factual knowledge. But for those of you out there who are saying, okay, this sounds great, how do I incorporate it into my classroom? So the question comes, what facilitates formative frustration? What are the details? And so we wanted to pull out some of the key things that you might utilize in order to incorporate into your classes to use this formative frustration concept to make the class better. And one thing is using or incorporating real research questions with unknowns. Another, as indicated in the quote by Anne, is rule breaking, paradigm breaking, things that don't fit the molecular standard paradigms, or, for example, molecular biology principles that are always taught in the textbooks as the baseline things, and requiring students to step back and actually look at multiple data points. And, the multiple data points, obviously, Justin talked about lots of different data points as well. And then not really touched on today, one aspect that we also, there's data for in the paper, was actually the value of dry lab research. And in fact, all the genome annotation work that we do, while some people incorporate aspects of wet lab, the annotation process is in silico, it's dry lab. And so the feeling is that, and in fact, the data bears it out, that this low cost, easy repetition framework gives students a longer leash to make mistakes and have this iterative frustration process of trial and error. And of course, the collaboration and a collaborative environment that mentors and allows people to get through this frustration is key. Now, it should be said, when thinking about these things to incorporate in a class that you might build, you got to think about some potential negatives, what things could make it go wrong. And so you want to try to think about things, making sure you avoid excessive frustration so that you help students work through things so you don't have student attrition. And so I talked about the process of creating a class. But to finish up and putting it in pictorial form, here's the last figure from the paper, which really sort of summarizes some steps that we might recommend for you taking into account into building a class like that. And the first step really is, try to create a supportive community. Clearly, we have the GEP as this very supportive community, but you can create that in your classroom with students and teachers and even TAs that help people through this frustration process. You also want to have a clear learning objective in the context of the GEP work. We have the goal of genome annotation, right? And then within that, you want to actually have a formative strategy that helps students and actually gives them stepwise lessons so that they gain the knowledge to build towards success. But at the same time, build in some frustrating situations that allow an iterative process that allows the students to learn and better understand the material through failure, which leads to them thriving and better understanding the information. And so with that, it's really important to thank our community for this. So first off, Sally Elgin, as Justin said, the founder and driving force of early years, and still a force to be recognized with the GEP. David Lopato, a key author, a senior author on this paper too, is our scientific assessment guru at Grinnell College. Laura Reed, who's now taken over the reins of leading the GEP at University of Alabama. And of course, this is a team of so many co-authors through the GEP that's put together this work. And none of this would be possible without the NIH and the NSF. And I would also like to step out and say somebody who's not listed on this, but another driving force, Anne Rosenwald, who's here today. We really, really appreciate all her efforts shepherding papers, multiple papers, not just this one, along in the science education community. So we owe her a big thanks to this, for this. And then besides that, we encourage questions. And also for those of you who think this GEP community would be a supportive and great framework to design courses that formatively frustrate your students and yourself potentially, but all in good ways, come join us. We invite you to talk, to ask more about the GEP itself.
Thanks, everybody. So we'll entertain questions. There's a couple in the chat already. It looks like Amy was going to tackle that one, the one about timeframes.
Right. I just wondered if we wanted to address it at the end. I think it's a really good question that came up while we were talking about the structure of the course, and Matt was speaking about considerations for implementing experiences that might allow for this formative frustration. The GEP has, and any of my colleagues here can chime in, but I'll just start by saying the GEP has members that use this kind of gene annotation and the GEP curriculum in a variety of ways, from really just learning gene annotation without actually delving into a novel research question, to people who make it a half a semester part of a course, a central part of an entire course that's a semester long. And I use the GEP materials with my independent study research students too. So there's a whole range. Some of the GEP's previous science education publications have looked at how time that students invest impacts their outcomes. So we have looked at that before, but just in terms of those of you that are interested in joining, there's really a lot of different examples of how people have used it. I don't know if Justin, Matt, Ann, you want to add anything to that?
The only thing I would add is we have a bunch of different curriculum that you can access on the website that's listed here. So some people actually don't get involved in the annotation part. We have a series of lessons that we call the eukaryotic gene modules, that allows students to learn how to use the browser and think about what a gene is and think about how you use the information that's on the browser to construct a gene. And so some people just use that, and that's all they do with their students. Some people use that as a precursor to them having their students do the full annotation with unknowns. I've tended to use it in an upper level bioinformatics class, and I just throw them in. I just say, okay, here's what we're doing. And I walk them through a couple of examples, and then I set them free on their own chunk of DNA. So people, Amy's absolutely right. People use it in different ways.
So I want to tackle the question about what happened to students who did not succeed, right? And did they appreciate the course? So I would argue, and maybe this is just my experience, my co-authors can chime in also, that students will almost always figure out some sort of gene model, right? And most of the time, they will come up with something that makes sense. Whether that gene model agrees, so we do a sort of quality control piece after a student submits a project to see how well it compares with another student who's doing the same project. But they will come up with some sort of a gene model. So I in terms of if they did not succeed and if they didn't appreciate the course, in my experience, students have always come up with something at the end. And so I feel like they, and students are going to like it or not like it, right? Or appreciate it or not. But I feel like at the end of a semester, it's not like you're categorizing them. Okay, these are the ones that made it through and these are the ones that didn't, right? I think there's always sort of some success that is made, even if it's little bits of success along the way. And how well the students appreciate the course, that I'm not sure if we actually, students usually say good things, but I'm not sure if we actually quantitated that information.
There is some evidence in the literature, which we cite in the paper, that talks about the benefits even if you don't succeed. I can't remember the citation off the top of my head. Amy, do you remember that?
I'm sorry, Anne, I'm not sure exactly I know what you're thinking. I'm not sure I know exactly what you're thinking of right at this second.
Well, I think it's in the discussion. I would have to go look, but there's some evidence there.
I would just follow up on the idea of succeed with how many of the classes are actually structured, and many groups organize things so that there's an end of semester research presentation or process. It depends on how much time you have to allocate to things, but this end of semester process and presentations, even if you weren't able to get as far on your annotation project as others might, in the end, I've felt, and I think a lot of others have the sense that that this seeing the group work and all the things that you need, the iterations in trying to solve a problem that you have to go through, the group, the supportive group, by seeing others, you see some successes that way. Now, it should be said that there are certain students in any class that say, this isn't my thing, right? And we could always argue that so long as you're not losing a certain segment of the population, student group on a regular basis, that in fact, giving them that frustration, especially if it's really helping them learn the scientific process, they may decide, well, okay, doing the scientific process isn't my thing, and that actually is a good lesson as well.
There's a question from Sarah that says about the intersection of diversity, equity, inclusion, and frustration. We didn't parse it out in this study by type of institution or representation in underserved minority groups. In general, though, there is data to suggest that cures are more inclusive than other kinds of tuition, and that it leads to better retention of students who are in underrepresented minorities. So the data that we have on that for GEP fits that model. There's a really nice study that came out in Cell Biology Education a couple of years ago that talks about the freshman research initiative at the University of Texas at Austin. And I can't remember the first author's name, but Aaron Dolan, Dolan, is one of the authors on that. And they did a case-controlled kind of match study for students who were involved in this freshman research initiative and students who were not. And the data, by and large, suggested that that all students had faster time to graduation, that they stayed in science longer, more, and so that really is, I think, the gold standard for thinking about issues of equity and inclusion in STEM education.
I think the other thing I would just add there, Anne, is that even though this report that we're talking about today didn't focus on details of demographics, where the GEP is really invested in continuing to figure out and ensure that this curriculum can work at a variety of institution types with a variety of students. And the other point would be that, as Matt mentioned, when establishing the student focus groups, right, effort was made to make sure those groups spanned institutional types. So not only do we have the demographics of all the students involved in the GEP at a given time period, but in the focus groups here, we really did want to try to assess students who are different institutional types. So that might be something to keep in mind just for this study as an example.
And something else to add to that, while we didn't look specifically at the frustration piece, we have been assessing the GEP students for years and years and years, and we assess them they take a quiz, right, so we can get at their student learning, and then they take a survey on their attitudes. And the GEP experience, for as long as it's been in existence, has been a positive influence on students, the students' survey results as well as the quiz results. And we've never found any correlation between a quiz score, for example, or a survey result and a specific type of institution, right? Undergraduate place, a research institution. So in that regard, in terms of student groups at different types of institutions, the effects are similar, right? In terms of type of student, we've never parsed it out that way. But, it seems from what we have been able to parse out, it seems to be affecting student groups equally, for whatever that's worth.
I'm just looking at that next question down in the chat. Maybe unless somebody else wants to say something more about the diversity, equity, inclusion piece right now. that next question, that's an anonymous question about thinking about this connection of GEP with frustration and the difficulty, it made me smile because, right, that is one of the challenges we're discussing working on this paper. For a while we even tossed around this term formative failure, right? But failure has such a negative connotation, right? And it's not failure forever. It's mini failures that create frustration and then ultimately seem to promote learning. But I do think that, what Matt said at the end about potential negatives, right? The challenge with any kind of work like this that's more open-ended is that it may provoke some frustration. And so I think that the support piece is really important. And we saw that in the comments, right? If students feel supported and they can work with each other or a TA or a faculty member, I think many, if not all of them, really do get something out of it. But I think it would be very important to be aware to gauge that, right, when setting up a class. I think going into it knowing that you want to be aware of take a pulse of how the students are doing and provide that support to prevent it from going into a place where frustration isn't positive, I think is an important thing to keep in mind.
Yeah, I mean, I guess I would summarize what Amy said by saying we want it to be hard, but not impossible.
So following up on that question, I think it's important that the text of it said linking GEP with frustration. And certainly GEP curriculum, which can be incorporated in classes from freshman to upper level, depending on how people choose to do it, it's hard, right? And that's Justin described how hard the process of annotation and how sometimes frustrating it can be. However, linking, it's also important to note that this doesn't just have to be GEP and frustration, right? You in other courses, other questions could incorporate this concept of frustration, whether it be with GEP material or not. And the idea is to just simply have materials that help and foster environment that helps and guides students, but also allows them to hit snags along the way so that they have to work through them and come out on the other side the better. And the challenge is, whether it's GEP or other, finding that sweet spot between snags so people don't give up and enough material to help them move through so that they actually find the benefits and come out on the other side feeling like they've learned.
So Tammy asked if you could share an example of using GEP in Cell. So I've used it in Cell before, and basically we just do annotation. Mostly what I choose. So what Justin talked about was the original project where we were looking at what's called the dot chromosome, which is very heterochromatic, and there aren't very many genes on there. But in addition, we would also have control projects on another chromosome. And so I would always choose projects from the control chromosome because they had more things that you could relate to cell biology. Not that no gene is related to cell biology, but the idea that you might have actin or something on there. So it was easier to make the connections there.
Well, and now, right, that the projects are expanding, right? So one of the big projects is signaling pathways in Drosophila, and we're focusing on insulin signaling right now. So I know a lot of folks who teach cell biology or even biochemistry are really excited to use those projects because the context will match their course, right? So you can talk about signaling, and yes, there's an evolutionary bend to the projects that we're doing. But at the end of the day, we're focusing on signaling pathway members and whatnot. So I think it's now an easier way to incorporate the GEP project into a cell biology or a biochemistry course.
And I would add, actually, in the cell biology or biochemistry or molecular biology, whether you're thinking about when you're starting to think about gene structure, you might have somebody run out a gel and look at DNA on a gel, but but that's kind of intangible. Whereas if you're doing the annotation and actually going in and looking at the ACTG, the nucleotides, and seeing and touching those nucleotides in silico, but still touching them more than you mind to gel, right? It actually makes introns and exons and 5' UTR and various other aspects of gene structure potentially even more tangible than running out a gel and seeing it as an abstract band that you don't see ACTG, right?
So I totally agree with that.
I would agree with that.
I mean, so I have taught this, the GEP in a context of a lab course where the students would do the annotation. And then, so I'm a fly biologist, or a fly geneticist. So I would get the flies from the genome that we were annotating from, and the students would isolate RNA and they would do RT-PCR, right, to see if they could confirm that their gene model, right? And I would always tell them, all right, choose primers across an intron, right? And there'd always be some genomic DNA contamination. So they'd be seeing, different size bands, and they'd have to interpret that PCR result based on the gene model that they made. And that was always a really part of the course that the students really enjoyed, making that connection to sort of the real world.
And I would say also, in my experience with students, that they've heard about introns for a million years, and they think they know what it is, what they are, and how splicing happens. And it's not until they do this project that they realize they don't have the understanding they thought they did. So it's valuable in that respect too, because it really teaches them where the gaps in their knowledge are.
Tammy, I might be able to address your question because I've used GEP in an upper level genomics class where half of the semester they're essentially doing a GEP research project. It's a capstone class, and it really runs like a little mini independent research project. And then, as I think I might have said before, I also use it with my independent study students because I, like Sally Elgin, am interested in epigenetics. And so when I joined the GEP, I was interested partly based on my own scientific interests, separate from science education. The GEP has been great for both. So I think it works great. And you can feel free to reach me. You can find my email at Muhlenberg pretty easily if you want to talk about it more. I'm happy to answer more questions now, but I do that, I would say, fairly routinely, what you're asking about, Tammy.
So, and there's another follow-up question. Do you see the GEP work done by students reaching publication? And absolutely, we've had numerous scientific publications on this as well. And that's available on our website and PubMed cited and things like that. But actually, going back on top of it to what Amy said, the class that I teach is independent research project as well. It's an upper level course, and I incorporate half of it, similar to what Amy is, as genomics or genome annotation, but the other half is sort of what do you do next from genome annotation, and it goes back into sort of Drosophila genetics and genetic screen type thing, and so. But it can absolutely be incorporated into an independent research. One thing that I think is always interesting to think about when designing these things is the process and teaching things through. I've actually found that teaching it in a course is sometimes more organized than my lab independent research, where I have so many students at different levels of understanding. If the course, they're all sort of starting at the semester at a certain time, and you're stepping them through the framework of the things that they need to understand, where that's quite difficult for me to do in my independent research lab, where I have freshmen, sophomores, juniors, seniors, all at different levels of understanding. So in that respect, it's actually sometimes maybe even better than a lab research experience.
Amy, speaking to the publication things, can you talk about the micro-publications? I think you've been involved in that more than anybody else at this point.
Yeah, I think I could talk about that, Anne. So as Anne said in the chat, with the insulin signaling pathways project that Justin was referring to, one of the newer scientific questions the GEP is engaged in, we started to try a micro-publication format with micro-publication biology, right? Where instead of only having a big summative paper that maybe, for example, looks at evolution across the insulin pathway, we're actually preparing micro-publications now that report gene annotation for a single gene in a single species. And my students have been working on the Pathways Project since last spring. And I think as of yesterday, maybe we've looked at 8 manuscript drafts for micro-publications. So I'm really excited about that as another way to enhance what we're doing in the GEP, because I think it's going to allow more students to reach all the way through and be really intimately involved in what it's like to look at a manuscript draft and the publication process. On our scientific publications, student co-authors were always asked to read the manuscript and offer comments and give their approval. But when you have only maybe 1 to 3 student co-authors on these micro publications, they're just, I think they feel more ownership. It's been very positive for my students, I will say.
And there was a question in there, any involvement with C. elegans and other, and Anne noted that largely doing fly stuff at the moment, but we actually have a project. There's two kind of ways of nodes of getting involved with this. You can be involved in the assessment and the research end and taking projects that come from above and helping to complete them. But there's also interest in bringing in people who have scientific projects, scientific collaborators that want to have a framework question. And in fact, there we have a gene on-ramp software where in fact the genome of interest can be plugged into that and brought into the annotation program in order to help students use that. And so that's not just Drosophila specific. I think in fact there's been a group in Puerto Rico trying to do some work and put that into gene on-ramp with the Puerto Rican parrot, right? But it's certainly not Drosophila specific. And the G on-ramp software is designed to help draw people in who might have other scientific questions, not just Drosophila based.
My sense, Fernando, was your question was more about the micro-publications.
I mean, I will say that I think, working in one of the species that has a strong community resource, my experience, because I've also published in micropublication biology in Arabidopsis, which is my wet lab research model. And I think having a good connection with the community resource, like C. elegans and obviously Drosophila do, is important. So, Fernando, in that sense, C. elegans would be great because worm base is so well established, just like fly base. And I think that would be an easy connection on the micropublication front. With the Arabidopsis, we got it worked out. It's a little trickier. And I think that would be the same for other species that don't have a community resource that's already linked up to micropublication biology.
I think worm base already is connected to micropublication biology, right? It's just correct. We don't have any scientific partners in the GEP that are interested in C. elegans projects right now. So we wouldn't have an opportunity to do C. elegans things. But as Matt said, we're always looking for scientific partners who have interests in, in using the GEP framework to answer one of their specific scientific questions.
All right. So with that, thank you for attending.